Imagine a car. A well-functioning car needs two key systems, an accelerator to move it forward and brakes to stop it. Now let's say the accelerator is stuck in full throttle and the brakes are completely broken. The car should be unstoppable, right? But here's the twist. Even with the accelerator stuck in overdrive and no brakes to stop it, the car can't move without fuel. The fuel and how it powers the car's engine is a crucial part of the story. Now let's translate this metaphor into cancer biology. In this car, the accelerator represents KRAS, an oncogene. This gene drives cell division and growth just like pressing the gas pedal moves the car forward. In mutant cancers, KRAS is locked into the on position, pushing cells to divide uncontrollably. On the other side, the brakes in our metaphor represent the TP53 gene, a tumor suppressor, and its job is to prevent uncontrolled cell growth, much like brakes would stop the car from just speeding out of control. When TP53 is mutated, the brakes are broken, and the uncontrolled growth goes unchecked. But let's focus on this fuel. This fuel is the metabolism, the network of chemical reactions that provide energy and building blocks for cells to grow and survive. Here is where things get interesting. Cancer cells don't use the fuel the same way healthy cells do. They're like a car that requires specific fuel, perhaps diesel instead of gasoline, or maybe they consume much more fuel than a normal car. And cancer cells with TP53 and KRAS mutations are especially reliant on their altered metabolism, just to support their rapid growth and survival. The key idea is simple. If we can identify what cancer cells need to survive, things that normal cells don't, or at least not to the same degree, we can develop therapies to target these vulnerabilities. In my research, which focuses specifically on lung cancer, we've seen that one of these key building blocks, purines, play actually a central role in several important cellular processes, such as DNA synthesis and repair. And what's even more interesting is that cancer cells with both TP53 and KRAS are actually relying a lot on these purines. We are trying to understand why this is the case and how to target these metabolic dependencies by repurposing some existing therapies. By cutting off the cancer cells' unique fuel supply, we could slow their growth, halt their multiplication, and even kill these cells without harming the healthy cells that don't share these extreme metabolic needs. This research is particularly important because lung cancer affects millions of people worldwide, with 2.5 million new diagnoses every year, and around 10 to 15% of these cases will actually involve both TP53 and KRAS mutations, making this approach potentially transformative for a large number of patients.